talk. The series is being recorded and will hopefully appear on YouTube for posterity. So this has been going on for the last year, and hopefully we're going to carry on beyond lockdown. So if anyone has any great suggestions for speakers, then let us know. I'm going to hand over for today's introduction to Kurti.
Yeah, it's my great pleasure to introduce Uri. So Uri did his PhD from NIH with Bechara Kacher, and then his postdoc with Jennifer Lippincott at NIH and Genelia Farms. I usually tell some short stories, people whom I know personally. So one of them is when I was visiting Uri for a position in Salk in 2000, I think it was 16 or 17, I forgot. Uri asked me which is your favorite denoising paper. And I accidentally said one by Peter Carton and Jerome Boulanger. He's at MRC. He usually comes to our talk. I'm not sure if he's in the audience today. And then Uri said, oh, that's also my favorite paper. I don't know if Uri really meant it or not. It was PNAS 2010 paper. But so since then, Uri has focused a lot on denoising, and now he has jumped to machine learning, deep learning kind of stuff. And some new exciting areas are hearing loss. Mitochondria is his all-time favorite. So hopefully he will tell us more about his research. Yeah. So Uri, now looking forward to your talk.
All right, I had forgotten about that. Thank you very much. That doesn't mean much that I had forgotten, because, you know, it's 5 a.m. over here. I don't remember anything. I barely remember who I am or where I am. But I do know that I'm really excited to be here. I like to complain, so I'm going to complain about 5 a.m. for the rest of, you will hear about it for the rest of your life. I guarantee you. But the reality is I'm very honored to be speaking here. I'm very excited. And thank you very much for having me. Really appreciate it. So I'm going to jump right into it. I'm not going to talk about hearing loss today. I'm going to talk about a lot about mitochondria. And I'm going to talk about some tools that we built to study mitochondria, and actually give you a little bit of biology as well to illustrate how these tools have actually allowed us to learn new things about biology. So it's not purely a tool talk, so to speak. So just a brief introduction. Maybe some of you have heard before that mitochondria are the powerhouse of the cell. Maybe some of you have seen these nice cartoons where you've got, you know, an inner membrane and an outer membrane of the mitochondria. Maybe you've even been lucky enough to see a nice electron microscope image of mitochondria. But what less people tend to know is that mitochondria are actually incredibly dynamic, and they're constantly moving throughout the cell, dynamically repositioning themselves and constantly fusing and dividing. So in this video, you can see in a single cell, this is one cancer cell, and it's got hundreds of mitochondria. And you can see in one place of the cell, you're going to see fusion, and another part of the cell, you're going to see fission. And that's when mitochondria come apart or come together. This is essential for their health and for the survival of the cell, and ultimately for our survival, because that is how mitochondria constantly compensate for damage to their DNA, and they constantly swap DNA with one another and make it through the brutal existence of life. So we have a vested interest in understanding how these dynamics are regulated. And as you can imagine, if you mutate any of the proteins involved in these dynamics, you can get a whole slew of diseases, ranging from encephalopathy, Charcot-Marie-Tooth disease, I'll be talking about later, and then Parkinson's. So anything from neurodegeneration to cancer to metabolic disorders can result from perturbed mitochondrial dynamics. So how are mitochondrial dynamics regulated? Let's focus on fission. So what's been known for decades is that there's a protein called DRP1, it's a large GTPase protein, forms rings around mitochondrial the fission site, and then when it hydrolyzes GTP to GDP, it switches from a 110 nanometer ring down to a 50 nanometer ring. But we have a problem. which is that DRP1 rings in their largest circumference form a 110 nanometer ring, whereas we know that mitochondria can be usually between 250 to 450 nanometers. So how do you fit a 110 nanometer belt around a 350 nanometer, again, it's 5AM, 350 nanometer waist? How do you do that? So really key insight came from Gia Volz's lab in 2011, where they were imaging mitochondria in live cells, and they saw that there are these pre-constriction events that occur prior to fission. And what they saw is really remarkable, is that the endoplasmic reticulum forms tubules that will wrap around mitochondria at these fission sites. So their insight suggested that the ER is wrapping around and squeezing down mitochondria prior to DRP1 coming in and forming that ring and performing the final fission event. Very cool. But there's a problem, which is that the ER is a 50 nanometer tubule. And how can a 50 nanometer membrane flexible tubule be squeezing down what is ultimately a bacterial cell? Something must be exerting that force. And when you think about force in the cell, very often the actin cytoskeleton comes up. And there is plenty of evidence that the actin cytoskeleton is important for mitochondrial fission. Tons of papers where they knock down actin regulatory proteins, INF2 is going to become very important later, or they use drugs to disturb actin polymerization, or myosin 2, and all kinds of other different perturbations of the actin cytoskeleton ultimately result in perturbed mitochondrial fission. For my postdoc with Jennifer Lippincott-Schwartz, what we showed is that there's an actin nucleating protein on the surface of the mitochondria called Spire1C. And there's, of course, this INF2 protein on the ER. And what we found, or what our model showed, all of our data pointed towards this, is that Spire on the mitochondrial outer membrane and INF2 on the ER interact. And at ER mitochondria contacts, that results in an upregulation of actin polymerization. And it's that burst of actin polymerization at ER mitochondria contacts that provides the force necessary for constricting the mitochondria, thereby leading to fission. But what killed me was I never had an image of actin clearly accumulating at an ER mitochondria contact site. And the reason I never got that image is reflected in this image right here. If you stain actin in the cell, you end up with this huge, beautiful, bright, organized network of filaments and bundled filaments all over the cell. And then you look at the mitochondria, and the mitochondria all over the cell. And you have to squint and try to convince yourself that maybe this actin right here is associated with that mitochondria. Maybe it's not. It's really hard to tell. You don't have the resolution, and you don't have the signal to background to really tell which actin filaments are associated with mitochondria and which ones are not. And that's because most of the actin in the cell is extremely bright and extremely stable, whereas the actin involved in mitochondrial fission, which can occur over seconds, is very small and very transient. So it's like trying to see the stars in the middle of the day. So I was inspired by a paper that I came across in a seminar where someone was studying nuclear actin, which is a similar problem. There's much less actin in the nucleus than in the rest of the cell. And the way they were able to visualize the nuclear actin was by using an actin nanobody that they had slapped on a nuclear targeting sequence. And using that, they were able to then actually visualize changes in actin cytoskeleton in the nucleus over time. And I was blown away by these images and by the signal to noise. And I thought, what if we did the same thing? What if we took off that nuclear targeting sequence and we put on a mitochondria targeting sequence or an ER targeting sequence? So that's what we did. We call these actin chromobodies. And what it is a targeting sequence that localizes the probe in the C-terminus, like a tail-anchored sequence to either the ER or to the mitochondrial outer membrane. And then we've got a fluorescent protein, so you can see the thing. And then you've got an actin nanobody that actually binds to the actin filaments. And a model for how this works is it diffuses along the membrane of whatever organelle you've targeted it to. And we also always use a control probe that has a different color fluorescent protein, and it does not have actin binding activity, but it has the exact same targeting sequence. And so both probes will diffuse freely along the membrane, except when actin comes within about 10 nanometers of that membrane. And when actin filaments come along that, near that membrane, then the actin nanobody starts to bind to it and it will start to accumulate. And the more actin filaments you have next to your membrane, the more actin nanobody you'll have, and that will result in an increase in fluorescence intensity. And we validated this model using fluorescence photo-bleaching experiments that I won't go into. But lo and behold, that is what we saw. You can see on the left our actin AC mito probe, that's the nanobody targeted to the mitochondrial outer membrane. You can see how it has these hotspots of increased intensity and decreased intensity, whereas our control probe, identical, except it doesn't have actin binding activity, is smooth as butter all along the mitochondria. And then we have a mitochondrial matrix marker. And what we could see when you zoom in is that you have these hotspots where you have increased actin filaments along different regions of the mitochondria. And this blew me away because I've never seen anything like that with a pan-actin probe, like colloidin or Lipact or whatever. This is something that I'd never seen before. I was very excited by it. And by the way, we quantified this hotspot, this accumulation stuff, as coefficient of variance. So the more variant the signal is along the mitochondria, the higher the coefficient of variance will be. We saw the same thing for the ER. So when we put our ACER probe in cells, you could see that there were hotspots in different regions of the cell, whereas our control ER probe was smooth as butter all along the ER. And what was super interesting is that a lot of these hotspots tended to be right there at ER mitochondrial contact sites, which is exactly what we had in our model here, but never managed to get an image actually showing actin accumulating at ER mitochondrial contacts. So this made me super duper happy. And now we know we have a tool that works really well for looking at organelle associated actin, and we're ready to go to town. So we made this nice visual showing that we can now see stars in the middle of the day. So let's do some live imaging. One thing we saw was that if you image mitochondrial fission events with our probe, invariably we see our AC mitoprobe accumulating specifically at these fission sites, which again was something I couldn't clearly see with any other type of actin probe. Same thing for the ER. We would see that ER associated actin accumulates specifically at mitochondrial fission sites. And we quantified to show that this isn't just random signal popping up at fission sites by looking at the total amount of ACER along mitochondria or AC mito along mitochondria. And we could see that the probe really covers about 15% of these organelles, but it's 100% of the time at the fission sites. So it's not a coincidence for the skeptics in the crowd. Now, Gia didn't stop with her ER mitochondrial work. She later showed that the ER also accumulates at endosome fission sites. And then later in a more recent paper in Science, she showed that the ER even accumulates at membraneless organelle fission sites. So this seems to be a universal mechanism where the ER regulates organelle dynamics. Knowing that, we thought maybe ER-associated actin could be involved in all of the organelle fission events. And that makes sense because if you image all the organelles in the cell alongside the ER, you'll see that the ER is pretty much always touching all of the organelles. So we decided to look with our ACER probe at all the organelles. lo and behold, we saw that ACER accumulates pretty much always at pretty much all of the organelle fission events. We looked at endosomes, we looked at peroxisomes, we looked at lysosomes, and even the Golgi, which was the hardest one to see. But in all the ones that we looked at, we could see ACER, in other words, ER-associated actin, was accumulating at organelle fission sites. So that sort of begs the question, is there a specific ER-associated protein that might be responsible for that? And if you've been following the literature, this protein called INF2 is a #1 target. So INF2 is an ER-anchored formin protein, and formin proteins polymerize actin. And here you can see INF2 is all over the ER, and it makes sense to look at it. I should point out that INF2 is also implicated in Charcot-Marie tooth disease. So there are so-called dominant activating mutations in INF2. So most, if not all, formin proteins are auto-inhibited. Their N and their C termini bind to one another, and that turns it off. And usually some other protein needs to come along and bind to its N terminus to activate it and basically cause it to remove that auto-inhibition and then be able to polymerize actin. And if you mutate that N terminus that's involved in auto-inhibition, then it's just on all the time, constantly making actin, and that leads to problems. And in the case of these patients, it leads to Charcot-Marie tooth disease, which is a so-called length-dependent neuropathy, which means that the longest axons degenerate the fastest and the most. So in the case of patients, that means that their feet, because the longest axon in your body is going all the way down to your toes, degenerate first, and then their hands, and then in extreme cases, sometimes the eyes, sometimes the ears. So one hypothesis for how Charcot-Marie tooth disease works is because these neurons are so long, because these axons are so long, they're going to be particularly sensitive to the mobility or any defects in mobility of certain key factors, such as organelles. So that all leads to our model for how INF2, this ER-anchored formin protein, could be involved in disease. And the idea is that it's constitutively active, which causes excess actin to be forming. And in the case of this ER-anchored formin protein, excess actin is going to be forming on organelles. And that excess actin could lead to excess fission, but it will also lead to immobilization of the organelles because you're forming this cage around the organelles. And there is data to support that. So Harry Haig showed in 2013, a beautiful paper in Science, that if you knock down INF2, you get reduced mitochondrial fission, whereas if you express a constitutively active mutant form of INF2, you get excess mitochondrial fission. They also showed that this constitutively active mutant reduces organelle mobility. They just remain frozen in place. So knowing what we know about ER-associated actin regulating all these other organelles, we hypothesized that maybe INF2 is also involved in regulating these other organelles. And lo and behold, that is what we saw. So if you knock out INF2, endosomes start to become really long and tubular, indicative of reduced endosome fission. And we were able to rescue that phenotype by introducing the ER-associated INF2 with a GFP construct and reverse the phenotype back to a normal fragmented endosome phenotype. Similarly, for lysosomes, if you knock out INF2, lysosomes start to get bigger. Whereas if you reintroduce INF2 back in, you can rescue that phenotype. So again, larger lysosomes, larger endosomes, larger mitochondria, all when you knock out INF2. So then we decided to look at mobility. And normally endosomes are dancing around the cell like crazy. When you knock out INF2, they dance around a little bit even crazier. And when you express a constitutively active INF2, the endosomes become locked in place. Same thing for lysosomes. Lysosomes love to bounce around on microtubules, and when you knock it out, they still party hard. But when you express this mutant INF2, they become fixed in place. So here we're converging on this sort of conserved mechanism where this ER-anchored formin protein is regulating the dynamics of not just mitochondria, but also endosomes and lysosomes. Now, to show that this was actin dependent, we looked to see if we could recapitulate that phenotype with latrunculin B. Latrunculin B is a drug that depolymerizes actin, and we did. So when you perturb actin polymerization, you get larger endosomes and you get larger lysosomes, similar to what happens when you knock out INF2. So next we decided to get right to the source and look at patient-derived cells, patients who have mutations in INF2 who have Charcot-Marie-Tooth disease. What we saw in these patients was these blobs of actin accumulation throughout the fibroblasts. And when we look closely, we could see that there were organelles trapped inside these blobs, showing that when you have these mutations, that you get excess actin accumulating around organelles. So next we decided, since this is a neuropathy, not a fibropathy, if that's even a word, we decided to look at neurons. So we took the patient-derived fibroblasts, and with the help of Rusty Gage's lab at the Salk Institute and Uri Manor, who's a superstar postdoc in that lab, we converted those fibroblasts directly into neurons. And what we could see was, perhaps unsurprisingly at this point, in the patient-derived neurons, the mitochondria hardly move. And the same thing for lysosomes. So here you can see in a control age-matched control-derived neurons, you can see the lysosomes dart around happily, whereas in the patient, the lysosomes stuck. And we next wanted to see if this is actin-dependent. So we treated the cells with latrunculin B to depolymerize the actin, pretty low dose, and we were able to restore the mobility of those lysosomes, showing that it is actually actin that's perturbing the mobility. So I'm going to bring us back to our nanobody probes. If it is true that it is actin that is perturbing the mobility, you might actually see that with our AC mitoprobe, and that is what we see. So if you look at this chronograph here, you can see that there's a lot of vertical lines. Those vertical lines are mitochondria that are not moving. But you'll also see some streaks, diagonal, more horizontal. Those are moving mitochondria. And in this middle row, you can see the AC mito signal. It's all vertical, which means that the AC mito is associated with the not moving mitochondria, whereas the moving mitochondria don't have AC mito associated with them. So this shows that it's association with actin that actually docks the mitochondria. Now, I've been showing you lots of images of mitochondria organelle dynamics. They look pretty, but what's been swept under the rug by myself and many other people in the field is that imaging live cells with high resolution is pretty stressful. You have to use an incredibly powerful laser with incredibly high doses to be able to see what we're showing you. And what people don't like to talk about, but is super duper important, is that if you do that for too long, you're going to change the cell. You're going to actually perturb the system you're trying to study. You are no longer studying biology. You are studying culinary science, because you are literally cooking your cells. So here's an example of that. Here's a movie of mitochondria in a cancer cell. And what you could see, I hope, over time, is that the cell's becoming darker, it's bleaching. Everyone's familiar with bleaching if you've sat under a microscope at a fluorescent sample. But what you'll also see is that the mitochondria are starting to swell and fragment. They're undergoing stress-induced fission. So ironically, we're inducing the very process that we wish to study. And this is unacceptable. So one way with a point scanning confocal, high resolution point scanning confocal, to get around this is to just image fewer pixels and give it a lower dose. The problem is when you do that, then your image isn't nearly as clear and you can't see what's going on anymore. As you can see in this movie on the right, we're imaging with 16 times fewer pixels, 16 times lower laser dose. But now you would be hard pressed to identify, let's say, a mitochondrial fission event. Whereas again, on the left, we're imaging with max resolution, but the cell is bleaching over time, and you're probably inducing a lot of the fission that you're witnessing. We have a similar problem with speed. So if you want to image at max resolution, you need more pixels. It takes longer to image all those pixels. And as you know, we like to study organelle dynamics along neurons, where the organelles are moving very quickly, very far. And if you do that at max resolution, you're going to have a very slow frame rate, and it's going to look like the organelles are hopping along the axon. And you're not going to be able to capture subtle cues like acceleration or deceleration as the mitochondria move around. Whereas on the right, we can image faster with lower resolution, but now we can't resolve the individual mitochondria as they pass by one another. And how are you going to track them? That's reflected in these kind of, oops. Sorry. So that's going to disturb our dynamics. I'm sorry, I'm skipping this slide. I meant to delete this because I'm trying to keep it in 45 minutes. So what I'm illustrating to you with these problems with speed and with toxicity is what we call the triangle of compromise, which is when you sit down in the microscope or with a camera or any measurement device, you often have to make compromises. Do you want more of this or do you want more of that? And in the case of microscopy, it's do you want more speed or do you want more resolution or do you want more sensitivity, i.e., do you want to be able to image with lower laser dose? And the laws of physics dictate that it is impossible to optimize and maximize all of these at the same time. So I was inspired when I saw a article in a photography blog. I used to have a photography business, and I don't have time for that anymore, but I still read about photography. And what they had done in this article was they used deep learning to restore low resolution images to high resolution images, low quality images to high quality images. Now, I have been familiar with deep learning for a while because we used it for segmentation of our electron microscope images. And many of you may be familiar with, for example, self-driving cars, where you might have a camera view and you need to be able to say, that's a car, that's a street, that's a tree, that's a pole, that's a bicycle, that's a person, you know, that's a baby in a stroller, or whatever. So deep learning has been around for a while for computer vision, but I had not thought of it in terms of image restoration or denoising, as some people call it, or super resolution, as other people call it. So I was really excited by this and wondered, can we use deep learning, super resolution, to restore low pixel resolution, noisy images that we acquire in our microscope? Because if we could, that opens up whole new opportunities and capabilities for our microscopes. So this is the approach we came up with. We first tested this with electron microscope data, and we got a hold of 160 gigabytes of gold standard, super high resolution electron microscope images from our collaborator, Kristen Harris. And then we used a highly sophisticated technical device that is, formal name is the Craftify. And what the Craftify does is it takes really high resolution, high quality images and makes them look crappy. Basically decreases the resolution and injects a ton of noise. Now we've got image pairs of high and low quality images that we then trained our deep net to learn how to go from the crappy back up to the high. And then we acquired real-world low and high resolution images on our microscope of the same sample and to test whether our model was making reasonable predictions. So you may ask, why Craftify? By the way, I should point out that this paper is now out in Nature Methods. Came out in March 2021. I should update. So why Craftify? So if you wanted to acquire enough training data, and you need a lot of training data to teach a deep net how to transfer an input to an output. If you wanted to acquire real-world high and low resolution images to train a deep net, you are in for a world of pain. First of all, to get the amount of data that we had would have taken 480 imaging hours, which would, in our highly subsidized core, cost us almost $20,000. Second of all, that's assuming it goes perfectly. Now, when you image with an electron microscope an ultra-thin section, you're actually damaging that section. It's becoming distorted. It's undergoing nonlinear distortions. And also, things are moving, right? And now you have to get two images of the same thing that are identical somehow, and then you're supposed to align them and then train a deep net. And if you want to do that with 80 gigabytes of data, you might be spending a lot of time getting that data. Whereas, if data like the data that you want exists already somewhere in the ether or in a collaborator's hard drive or wherever, you can just take that data, and if you can engineer the proper Craftifier for your data, for your purpose, you can just create these semi-synthetic training data and then train your model. And if you've got a computer like ours, only takes 2 hours, only costs $16. So that's why we use the Craftifier. This is just an example of the output. So on the left is a low resolution image that we acquired in the microscope. On the right is a high resolution image of the exact same sample that we acquired in the microscope. And in the middle is a deep learning output. And you can see that these presynaptic vesicles, those are those little circles here, that are pretty much very difficult, if not impossible, to detect in the low-res image, are now popping out as clear as day in the deep learning output. And you can look at the ground truth in the high-resolution data to know that it's not just making up nonsense out of nowhere. And I don't have time to go into all the details, but we did validate our results with, quantitative metrics, pixel-wise metrics, as well as segmentation of the presynaptic vesicles to show that it's actually predicting real things. So then we did the same thing for our live, bringing us back to mitochondria. So on the left, you can see the low-resolution microscope image that we acquired. And on the right, you can see the output of our deep learning model. And looks pretty good. We've gotten rid of all the noise, and the mitochondria are nice and sharp. But there's a little problem, which is that when you play the video, there's a lot of flickering here. And that's because it's doing the best job it can to predict exactly where and what the mitochondria look like. But there's a lot of noise. And every single frame, the noise is different. So every single frame, the prediction is a little bit different. And this results in this unfortunate flickering effect, which is a nightmare for studying mitochondrial vision, because every single frame, a mitochondria is joining or breaking, and that's not real. So we needed to fix this. So what we realized is that since our training data and our testing data is video, we have multiple time points. And therefore, we could make a multi-frame deep net. And what we did was we took T-2 all the way to T+2 time points. So 5 time points from each video, and use those 5 time points to make a better prediction for the center time point. So this is kind of like a rolling average, which some people use to get rid of noise. But instead of just averaging and blurring all those 5 time points together, which will cost you in spatial and temporal resolution, we're just taking those 5 time points to make a better prediction in our deep learning model. When we do that, we get rid of the flickering. And in fact, you can tell, and I hope you can tell, I don't know if you can tell, that the images are even sharper, because now we have more time points. We can make even better predictions for each time point. And not only did we get rid of the flickering, we actually have higher resolution than we did with the single frame model. And that ultimately allows us to detect mitochondrial fission events that would otherwise be nearly impossible to detect in the raw data. Similarly, we can now image ultra-fast time lapses of organelles moving in neurons, which would normally not allow us to resolve individual mitochondria passing each other. But after processing with our deep net, we can now see mitochondria passing each other. We can detect mitochondrial fission events and have lots of fun. And just a quick note on one thing we're working on next. The way people in neuroscience studying organelle dynamics in neurons traditionally analyze organelle mobility is with these so-called chymographs. So you draw a line along the length of the axon, and then you look at the vertical versus horizontal lines, and you get a sense of the mobility. And then if you want to actually compare all of the different organelles, what you need to do is draw a line along each one of these, and then measure the dynamics of each one of these lines. And that is a nightmare. So we're using now machine learning to try to help us get out of this problem so that we can do more high throughput imaging. So one thing we're doing is we're using Elastic software to be able to at least semi-automatically segment all of the individual organelles. So it's easier to get the individual tracks of each organelle. But we're also going further and decided that, once we have that, I don't know if we need chymographs anymore. So instead, we're actually doing just actual individual organelle tracking, and then we can pull out the individual path details. And using these approaches, we can now do high throughput quantification of organelle mobility, bringing us back to looking at patients versus age-matched controls. We can now quantify the differences in mobility with higher throughput. And what you're seeing here is something called a super plot, where the individual dots are individual organelles. The little dots are individual organelles. The big dots are the averages per cell, and each color is a different cell. So you can see where our end values are for each type of measurement, whether it's individual organelles, individual cells. So coming up next, we are still improving our deep learning model to increase the resolution even further using something called feature loss. We are making larger dimension models, including 3D. And something I'm super duper excited about is we're using deep learning to make predictions on what is happening in time from single time points, so that we can eventually simulate videos from single snapshots. And as you can imagine, a ton of amazing people were involved in all of this work. And I'm incredibly grateful and fortunate to have them as collaborators. It's a beautiful thing when you get to work with people from all over the world on these exciting things. And I want to thank my funders. We have a grant from the NSF NeuroNext program, headed by Kristen Harris. I have some funding for my hearing loss research, and the Salk Institute has been incredibly supportive of me as well. With that, I will tell you that we have even more funding and we have more slots. So if you know anyone who would be interested in working with us on any of these things, please send them our way. We have a beautiful team. It's like a family. These are amazing people that I'm so grateful to work with. If you ever heard of something called imposter syndrome, it's this slide that gives me imposter syndrome more than anything else, because I'm working with these people somehow. And anyway, it's really touching. So in any case, I will take questions and suggestions, and thank you for your attention. I don't know what time it is where you are, but for me, it's still pretty early. Normally, I'd still be asleep, so forgive me for my ramblings. And thank you.
Great talk. So while you can post questions, shall we do the quiz, Uri?
Oh, I forgot about the quiz. Yeah, give me the quiz. What I don't even remember what questions I put in the quiz.
Okay, Georgina, you make me host so that I can share the screen?
Yeah, you can share the screen. You could try. Does it work?
Yeah, awesome. So
Cool.
yeah. So let's quiz. So we have about 9 people, so we'll give a couple of minutes for everyone to join. Yeah, maybe we post the link again from the chat.
I love it. I love it. Okay, scientist power.
Okay, should be okay. I don't know how to go this. Okay, so yeah, the link is in the chat. So we have now 20. Awesome. And like last time, we will have the leaderboard in. So, okay, maybe 30 more seconds. So actually, this question is copied from Uri's Twitter. I saw one question, what gives people power? So I left one option in and changed rest of them. So we have about 35 people. That's nice. Still more people joining in. So I think 39 is a good number. We usually have 20 people. So yeah, mitochondria is still leading the race, though knowledge is quite good. So your mitochondria is finishing second now. So money, it's actually third now. Money is second. So yeah, so we will also have the leaderboard this time. So I hope you listen to Uri's talk, and the faster you answer, the more points you get. So what is the eternal triangle compromise for microscopic imaging? First option is sensitivity, resolution, speed, sensitivity, resolution, signal to noise ratio, resolution, precision, and speed. Wow, that's nice. And let's look who is leading. So we have Snuggles, Liz, Saint Elmo, Watson, pretty much close, like first 5-1. So we have almost a pretty close race. So top 5-6 people, the faster you answer, the better it is. So Snuggles snuggling ahead. So the question 2, I think. So what does Craftification approach stand for? So Uri had to slightly change some of your answers because they were quite long. So I had to remove so the first option is manually acquire crappy images under a microscope for testing. The second and the third and the fourth had digitally downsample. So I thought, Uri, no one would manually downsample these days. So I removed the word digitally. So digitally downsample high resolution images to low res versions for training. And so possibly you can read. And so let's see what's the right answer. I actually forgot what was the right answer. So, we have a new entry. WG and Liz are tied up on the first spot, and Wolfmeister, Kerry. I guess with the last question, everyone has a good chance. So my instinct says Liz is going to win. So anyone betting? So we should start some betting also, right, Uri? To make it more interesting. Liz, money is on you. What are benefits of the multi-frame design for live cell time-lapse imaging? So this is, I think, multiple answer questions. So yeah, increase both temporal and spatial resolution, increase temporal resolution only, spatial resolution only, reduce flickering artifacts. Wow, yeah. So first and the last one. So let's see. Looks like WG, Sir WG Grace has won the race. So yeah, congratulations. So Stephanie will give out the fold scope. I'm not sure if she's in the audience, but if you send the name to Georgina, Amanda, me, or any of the organizers, we will send you a fold scope. So that's the end of the quiz. And now we ask Uri more questions. So WG, if you are in the audience, please say hello. And
I think it's actually provided now by the Royal Microscopic Society. So I used to send them out from my little village post office, but I think now the Royal Microscopic Society has kindly agreed to send them. So they have my collection, but and soon probably it will be depleted. So there will be more to be.
so regarding question, how we do it is you get a chance to ask the question yourself. So in the order, let's say Piyush, then we have Elizabeth, then we have Liz, then we have Susan and Maria. So in that order, if you can unmute and ask and probe Uri as long as you want. He's now fully awake. His voice has reduced the flickering, the flickering in his voice. So
That's a lie. But yeah, I will do my best.
Piyush, are you still there or?
Yeah. Hi, can you hear me?
Yeah, yeah.
Yeah, great talk. I really enjoyed listening to you. So I was wondering, most of the dynamics that you show are in regions where the mitochondria is like exists as a singular entity. So if you would could talk about regions where mitochondria is aggregated, and yet we want to study dynamics there. So is it possible with the deep learning?
So this is really important. At least our deep learning is not really magic. It's not going to give you resolution that you could not acquire on your microscope in the first place. So for example, with an AriScan microscope, you don't have infinite resolution. So our training data maybe probably couldn't get all of the mitochondrial fission in aggregated regions. For certain processes, such as looking at mitochondria in aggregated regions, you need the resolution of an electron microscope. And so we will not be able to see things that we could not see with our training data. Maybe a STED microscope would have sufficient resolution for looking at individual mitochondria in an aggregated region. I honestly don't know. If it did, then if we could get training data from a STED microscope, then maybe we could train our deep learning model to be able to do that type of time-lapse imaging. Does that make sense? Did that answer your question?
Yeah, thanks.
I thought, Uri, you didn't like dead samples, but it seems you like them in case of STED. So the next question comes from Elizabeth. I meant, Uri, what survives STED bleaching, right? So.
Oh, the STED, yeah. But you know what? This is a great example. Like if you can get tons of STED images, let's say the early time point before you cook the cell, and you train a deep net from that, then you can just use a normal confocal at lower resolution, lower dose, and maybe you can get some really amazing results. I've been blown away by what you can do with deep learning, but still, it's not magic. It's not going beyond the training data that you gave it.
I see Elizabeth. Elizabeth, if you, yeah.
Yes, hi, thank you. Thanks for your talk, Uri. My question was, in your Craftify, do you really only change your resolution, or do you also change like the noise levels and like the orientation within the images, or for example, things like that?
That's a good question. Thank you. We actually spent a lot of time optimizing our Craftify, which seems like a strange thing to do with your time, but it was very important. Because if the Craftify doesn't work well, your model output isn't going to work. So what we did was we tried many different noise models. And if you look at our Nature Methods paper, you'll see Figure 1 has what we call a Craftify comparison, where we look at different types of noise, including no noise, which would be just decreasing the resolution, and looking at the output of models trained with that type of Craftifier. And what we found at the end is something called additive noise. Some people call it multiplicative noise, which I cannot say even when it's not 5 a.m. I guess it's not 5 a.m. 6 a.m. So, but basically what it does is it adds more noise wherever there is more signal. So if a pixel has a certain amount of signal, that pixel is going to get more randomness, and the pixels around it. I think we use a sigma of 3 or 5 pixels surrounding the center pixel. Depending on the amount of signal, you give more noise. And that works really well for microscope images, because as you know, in a microscope image, you're going to have regions with nothing and then regions with a lot of stuff going on. So having that additive noise really helped us give data that the model knew what to do with, which is interesting to me, because if you're a microscope nerd, you hear that there's Gaussian and Poisson noise, and therefore you want your model to have some sort of Gaussian and Poisson noise. But in reality, when you're training a model, you want it to be able to look at the structures of and the sort of signal distribution in your images. And additive Gaussian noise gives you that. I don't know if that answers your question.
Yeah, absolutely. Thank you very much. Thank you.
Okay. All right.
Next, we have Liz. Liz, I can see you if you, yeah.
Yeah, hi. Thanks for a great talk.
Are you the winner? Hold on.
No, I came second, unfortunately.
Right.
Sorry to disappoint you, Kathy. I knew you were rooting for me, but
Yeah, good that I didn't bet, but next time we will introduce some betting also.
okay, sounds good. Yeah, my question was really relating to sort of how these actin forces manage to constrict the mitochondria. Because if you have the force between the mitochondria and a microtubule, I could imagine it going in different ways. You could have some deformity, you could have movement. So I was wondering if there's a difference in the density of sort of actin nucleation sites between the two surfaces that causes this constriction.
Yeah, that's a really good question that we cannot answer with fluorescence microscopy, in my opinion. So we have been attacking using a couple different EM approaches to try to attack this problem. And one that I'm a big fan of is membrane extraction, where you can remove the membrane and stabilize with a cytoskeleton stabilizing buffer, and then you can perform electron microscopy on that. And Tanya Spitkina had a beautiful paper looking at the actin cytoskeleton and mitochondrial fission site using membrane extraction and EM tomography of platinum replicas. And there you can start to get a sense of what's going on with the actin filaments there. And then the other approach we were looking at was cryo-EM, which is very low throughput, to say the least. And Danielle Grotjan at Scripps Institute is making some headway on that. But the really short answer is I don't think we know yet. I don't know if I don't know if density as much as the geometry of the actin filaments is going to tell us what's special about the actin in these sites. So I don't think I answered your question, is because I don't have it. I really don't know. But I threw out what I knew.
Thank you.
Is Suzanne, so I can see her. So go ahead.
Hey Uri, that was great. Thanks. My question was, do you see differences in the actin on mitochondria when you've got divisions in the middle of mitochondria compared to pinching off at the ends? Because there's a Nature paper out saying that mitophagy related kind of divisions maybe don't have actin. And I guess I was going to follow up with how does it look like in the neurons? Do you see the divisions that lead to retrograde transport differentially not associated with actin?
Yeah, I thought maybe, yeah, I was wondering why you were asking that question. So we see actin at pretty much 100% of all fission events, which I recognize that kind of contradicts that Nature paper. In their defense, they didn't have our probes, so they weren't able to see actin as easily as we can. And you know, Suliana Manley is one of the best scientists in the world today. Okay, like I have no doubt that they did everything right, and I think, like I said, if you're using these pan-actin probes, you're not going to see everything. But it's not really a major point of the paper, right? Like if actin happens to be at these other fission events, it doesn't really change the major conclusion, so it's not a big deal. But we see actin at all fission events. The question about retrograde versus anterograde is interesting and cool. We do see ER-associated actin at fission events in the axon. I don't think we've tried to dissect anterograde versus retrograde because we've seen it at all of them so far. So, but I do like the idea of trying to identify differences between those. That's a really cool idea.
I mean, if you can track all your mitochondria with different rainbow colors, it should be easy now.
Nothing's easy, but and when we do our tracking, we're not looking at the other probes. We're imaging one organelle at a time usually. Sometimes we image 2 organelles at a time. And you know, with deep learning, we can now do, you know, multicolor imaging. We might be able to do it. We haven't tried. One little baby step at a time. But yeah, that's a good goal to aspire to.
Yeah. So the next question is from Maria Livia. I can see her. So go ahead, Maria.
Yeah. Hello. Super nice talk. Really appreciate it. I was wondering, yeah, it's a bit similar to the first question. I was wondering, even though you have let's say you have the resolution enough in the microscope to detect even very interconnected mitochondria, how do you analyze them? I mean, with the method that I'm aware of, it's with the thresholding, it's very difficult to separate one to another mitochondria if they're so interconnected. Do you think the 3D for sure will help, like by eyes? I mean, looking, okay, second slice, yeah, that mitochondria is still there. So, I mean, and then I had more, sorry, yeah. Then I have another question if possible.
Yeah, no, please continue.
Yeah, and then I was wondering, so we try to monitor contact sites, right, which are super fast. And yeah, as you said, either the resolution or the timing. And yeah, I was wondering, since the resolution that we have, at least with fluorescent microscope, it's not up to more than 100 nanometer. Correct me if I'm wrong. And the contact site, for example, ER mito, they are at least 20, 50, if we want to be less stringent, nanometer. How do we can believe that we are watching contact sites? I'm doing a project on contact sites, so I do believe I'm seeing them, but those microscopes, they don't have the resolution enough. And yeah, as you said, the stat for sure. And the electron microscopy is not live. So if you can help me out, thank you.
Yeah, it's a really hard problem, right? So the first question was about aggregates, which I think I answered before somewhat, which is that ultimately a lot of fluorescence is not going to give you the resolution to be able to see all the structures you want to see in a cell. I'm sorry, it's just not, you know, and what ends up happening is you have to ignore those regions. You can't interpret it. You don't know what's going on, so you look elsewhere. And very often we find, even with thick cells where you have all the time in the world to try to reconstruct and get your image, you still have to go to electron microscopy to be able to tell where individual mitochondria are, where they begin, where they end, and whether they're fragmented or tubulated even. Sometimes it's really hard. So, you know, there are higher resolution methods that I don't currently use or have access to that maybe would do a better job with them. maybe with some deep learning, we can get some actual live time-lapse imaging of those aggregates. But right now, it's just a big problem. And one way we solve the problem is by using bigger cells. You know, if you're imaging HEK cells, which are these small round beasts from hell, you're not really going to be able to see much at all. Whereas if you use a giant COS7 cell or U2OS cell, you might have a better time, which doesn't often solve the problem for a lot of people. Patient fibroblasts, it turns out, are pretty big and amenable for imaging, so those are nice. But if you want to image, let's say, mitochondrial fission in the neuronal soma, which is pretty tiny, you're going to have a really hard time, and it's just going to suck. And you might have to figure out a way to incorporate EM into your dynamic live data. So one thing we're working on is, we're doing this a lot now, we do movies, time lapses on the microscope, live fluorescent microscope, and then we fix it on the stage. And then we find the exact same cell in the electron microscope. And so we know what was moving and what wasn't, and maybe what underwent fission recently, what versus what didn't. And then we can look at it in the electron microscope and get more ultrastructured, higher resolution data out of it. And my long-term goal that I sort of alluded to in my last slide, in one of my last slides, is that we're going to train a deep net to be able to predict the EM detail from the video data and vice versa. So if you know something was moving in one direction versus another, and we annotate it as such in our electron microscope, eventually we'll be able to look at just the EM images and know which direction different objects were moving in. That's going to require a lot of training data, but we're working on it. The other question was about contact sites. So there's tons of papers, mine included, with really shitty data showing pixel overlap, and you call it a contact site. I've done it. I'm guilty of it, because there was nothing else. And I also have papers where we do EM and we look at contact sites. They also suck, to be quite frank, because what is a contact site? Are they really touching, or are they just next to each other? What's the distance that you decide? It's also a 2D image. You know, maybe you really need FibSim to look at it in 3D, and even then. So there's no easy answer. I mean, one thing is split GFP, some of these molecular complementary probes where you can put half of your GFP on one organelle, another half on the other, and then when you see a signal, that means that they're truly touching, they're within a certain distance of one another. That's a pretty cool approach, but then you have to worry that maybe you're inducing artificial contacts, and or maybe there was a contact at one point, but it wasn't supposed to last. But one thing we did in our recent paper that worked is we have a positive control. So there are these known organelle-organelle contact proteins like BAP-B and PTP15. And if you overexpress them, you really induce contacts and everything changes. So that's your positive control. And if you measure that pixel overlap in the positive control, and then you show, let's say, in a knockout condition or in some other condition that, you know, you've measured some difference, you can at least show that you've attained the sensitivity in your measurement to be able to measure differences, but it's still not perfect. That didn't answer your question either, I don't think. I have seen people use something called PLA, proximity ligation.
I'm
assay.
using it.
I've never used it. It looks cool. Every time I see it, I'm excited. I'm like, we got to do that
cool.
too, but I've never done it. Okay.
It's cool.
Right.
You have to take
cool.
along the negative control and everything because you see dots everywhere, but that's science.
Okay, good to know.
I'm also using the FEMP. That's also something else, a FRET-based probe with a ER protein and a mitoprotein, and also the DDGFP. So FibSim too.
That's better than split because you're not inducing like a irreversible interaction.
The
problem is every time I look at FRET data, I'm like, I don't want to do that. I want to find some other way because Fred is pretty noisy sometimes, right? You don't get a ton of resolution. Yeah. But
Thanks
cool.
a
It
lot.
sounds like you're doing a cool project. Yeah. Of course.
Thanks a lot.
So we have a couple of more questions, and also we are over time. So would you like to answer more, or shall we call it off?
I'm happy to answer more. I mean, look, I'm not doing anything else. 6:05. My family's still asleep, so actually, I'm happy to keep going.
It's actually, Uri, I realized in one of the previous talks, a postdoc from your lab, Ling Ying Fang, she gave a talk at Imaging ONEWORLD, and she called you the Jewish mother. And now I realize why she called you her Jewish mother. You really give a very long, nice explanations with melodious voice. Actually, this is the second time I fell asleep during an Imaging ONEWORLD talk, and I just woke up right at the question time. I don't know how my brain kept the time, but yeah, I think it's just because of your super melodious voice and nice explanations.
I'm trying to decide if that was a compliment or not. I'm gonna have to stew on that for a bit. I fell asleep during your talk. Insult. But it's because you have a melodious voice. Compliment.
But magically, I just woke up just before the quiz, so that's nice. Catherine, you are next, and then we have another question from Liz, I think. Yeah.
Hi. So I'm very curious now that you've got this really these knockouts and this information and way of probing and looking at these interactions. Does this protein work on its own or in concert with other proteins, or are you looking to see for alternative candidates and maybe using these tools? How might you do that or exploit this?
That's a good question. I mean, I don't think any protein works in isolation, right? So we showed in my postdoc that Spire is a protein on the mitochondria that can bind to INF2 on the ER. It turns out there are other isoforms of Spire that localize to endosomes, not mitochondria. So maybe there's a conserved molecular mechanism there. We would love to explore that. There's got to be other proteins as well. So one thing we're doing with our probes that I'm super excited about, but don't have anything to show yet, is as you saw, we can now label actin associated with different organelles. So what we're doing is we've slapped on an apex probe, so we can proximity label the actin, the proteins associated with the actin. So we will pull down the interactinome, that's what I'm calling it, forgive me, of all the organelles. So we'll know soon what are all the actin regulatory proteins that are preferentially associated with the ER versus the mitochondria, and even in the nucleus.
Could you explain what exactly Apex probe is in this context?
Oh, I'm sorry. Yeah. So Apex is it's a form of peroxidase. You may have heard of BioID or TurboID more recently, Apex. So all of these things, what they do is they sort of label around them in the proximity proteins that you can then identify by mass spectrometry.
So,
thank
Liz,
you.
Your next question is good, but I will jump to Abigail. She has some exciting question which I actually don't understand. So, Uri, the new Abigail, go ahead. I see you now. Yeah.
Oh. Hi.
Hi. Good to see you.
Thanks for getting up so early to give a talk to us.
You're welcome.
I'm right there with you. 8 o'clock here in Boston is already painful enough. I can't imagine 5 a.m. Great talk. Just beautiful stuff. I had a question about, a question and a comment, which is also a question. Are there, now that you have seen some of this detail on actin, and I'm wondering if there's any new insights for treatment for people who suffer from the Marie tooth disease. I mean, I don't know that you can just give someone a drug that will mess up their actin that might cause a lot of other problems. But do you think there's any light there?
Yes, I do. Your point is really well taken about, you wouldn't, if you gave a patient Latrunculin B,
yeah,
you should probably have your license taken away, right?
Yeah.
You know what, maybe I'll share my screen. That's okay.
Georgina, I'm not sure. Yeah, if you are still
yes, fine.
Okay.
All right, can you see my screen?
Yeah.
So Latrunculin B disrupts all the actin in the cells, right? That's what we call a bad thing. So one thing we're working on is we're making genetically encoded tools to disrupt specific actin pools, inspired by my friend Brad Zucchero, who developed something called DACTs, which is a peptide derived from gelsolin, which is an actin severing protein. And you express it in cells, and it depolymerizes all the actin. So it's a genetically encoded way to depolymerize actin. And his thinking was you can put it in a cell-specific promoter so that you destruct the actin only in a specific cell type. Now, as you can see in this image, maybe I need to make it bigger, you've still really messed up the cell. And here we have data showing that ER-associated actin specifically is involved in these organelle dynamics. So we decided to piggyback on our actin antibody idea and make organelle-targeted DACTs and see if we can disrupt the actin only in proximity of a certain organelle. And we have a bunch of data showing that it works while leaving the rest of the actin intact. So this is a cell expressing our ERDX probe, which we found does disrupt ER-associated actin, but the cortical actin is fine. So the cells are still healthy for the most part. And oh shoot, I don't have the movie here, but we expressed our ERDX probe in the patient-derived neurons, and we restore mobility. So the organelles are mobile again.
Wow.
Yeah, it's pretty amazing. Whether you would use that in a clinic as a gene therapy is an interesting question. I don't think I would, certainly not alone. These days, CRISPR is amazing. Most of these mutations are dominant active mutations. So if you can inject, if you're going to do gene therapy, I would probably try to correct that mutation, that mutant allele, or just remove it completely so that you only have the healthy allele. But it's possible that even if you do that later in life, that the organelles will still have so much actin around them that they're not going to move. So maybe some transient expression of our ERDX probe could dislodge the organelles and allow them to move, and then hopefully maybe recovery. This is me, a cell biologist, speculating on what you might do in a clinic. We need to do in vivo studies. So we're going to make a mouse that has the same mutation as our patients and actually try these experiments in the animals first. And then if that actually works, then maybe we'll think about something for the clinic.
That's fantastic. It's nice to see things that can have such a direct impact potentially. But
it means a lot to me. I love science, and I think it's a lot of fun figuring out problems and doing things, but I also feel some strange need to justify my existence by helping. So it's fun to work on things that actually have a disease relevance. I think feels good.
So I wanted to follow up on your comment that you said you were doing some fluorescent imaging and then fixing them in the dish so that you could track back. I don't know if you remember our conversation from what feels like an eternity ago. We are finally, hopefully this fall, assuming we get COVID restrictions, setting up a high pressure freezer that will be integrated with the ARRI scan, so we can do imaging and then freeze the sucker for anything else.
Can I come?
Yes. Yes. We were supposed to get this.
I want that thing.
pre-COVID, but it fell off the airplane and it never made it to the country. So we got delayed a little bit. I know, horrible situation, but the plane was not in the air. It was being loaded. So shipping.
You mean it literally fell?
Yeah.
Was that in Paris? Well, I mean, France.
Yeah,
was that in France?
Yeah. So the high pressure freezer from cryo capsule, it got dropped off the airplane, not by cryo capsule, but by the ground crew. So that's why we don't have ours yet, but we're hoping to go through with the install this fall. So we're always eager to work.
Amazing. Yeah, I keep banging the drum and telling people we need to get one at Salk, and we haven't gotten it yet, obviously. Yeah, that is going to be a game changer. Not having to inject formaldehyde into your samples while you're doing your imaging, being able to freeze and get that perfect vitrification of your samples. That's so exciting. That's so exciting. I want to get that. And then the other thing.
Uri, Georgina has given me a 5-minute warning from now, which was 2 minutes ago. And there's one more question from Liz, which is very interesting one, and I'm not sure if she's still there. If Liz, are you still around or? Oh, so go ahead, and then Georgina has to run to some other meeting. So sorry for that, but I hope, Abigail, you can get hold of Uri again, and since he's awake now.
Abby and I need to catch up. We need to catch up offline. It was really good to see you, Abby. Thanks. Liz, I see your question. How do you make sure you're not just training your neural network to reverse the specific noise effects of the Craftifier? In other words, you're training your model with artificial stuff. How do you know that it's not just reversing artificial stuff instead of real stuff? And that's where the hard work of validating your model with real world, low and high resolution images comes in. So we trained our model on semi-synthetic pairs that we used with our Craftifier, but we tested our model and we quantified the results with real data that we acquired low and high resolution. And again, the real value of the Craftifier is that you can get 80, 160 gigabytes of training data with high throughput, whereas if you try to acquire all that low res, but you can still acquire enough. You cannot avoid it. You have to get enough high and low res real world data to at least validate your model. Otherwise, you don't know what you're doing. You're in the dark, and you could be in danger of messing up.
Okay, sorry, the final warning from Georgina has arrived, and this is the awkward moment when we say bye. We don't have anything, but hopefully we will at some point give some small mementos, souvenirs to our speakers. Great talk, Uri. As always, nice listening to you.
Thank you so much, Kirti, for having me. It's always good to see you. I really appreciate it. Thank you, everyone, for your attention, and to the Royal Microscopical Society for having me. It's a huge honor. It means a lot to me. And if anyone has any more questions, feel free to email me. You know where to find me.
Yeah. Thanks, Uri.
All right.
Thank you.
Thanks, everyone. Bye.
Bye-bye.